Hi everyone, today we are starting Doodle Sheet part 4, part 2, which is the last Doodle Sheet you guys will do in my biology class. I'm so sad that I'm giving this lecture to you this way, but it is what it is. We're all just doing the best we can. So today we're going to be learning about genetic replication and genetic engineering. So, as you should know already, parents pass traits to offspring by giving them DNA. So your parents, each of them gave you a portion of their own DNA. So try and answer this. Box A, when did the giving of DNA happen? Okay, what had to have happened for you to get that combined DNA? Hint, hint, what's happened over here? How many cells did you have when your life began? And how many cells do you have now? What do you guys think? Okay, box B. Parents pass traits to offspring by giving them DNA. Do all of your cells have DNA? What do you think? Yes or no? What do we know about DNA in all of our cells? So write down what you know. Oops. What do you know about that? And what does that tell us DNA must be able to do? So I know I'm not giving you the answers to these questions. These are questions to help you think about what do you already know? What do you remember from the work you did prior to spring break when we were in distance learning? If you don't know, just write down what you think. That's okay. You can look back on this after this lecture and go, oh my goodness, now I know the right answer. Just write something in each of the boxes. It's perfectly okay for box A and B. Okay, please take a second and watch this video. As you know, if I play it, you won't be able to hear it. So please pause, watch this video. It is very informative. Okay, so model of DNA replication. Okay, how does DNA make copies of itself with all of your cells? So watch this video in box C. This filled out for you. So the process has been diagrammed in your doodle sheet. So to help you understand this image in your doodle sheet, please watch this video. I know this is annoying. I'm sorry. I wish I can play this for you. It doesn't work that way. Okay, so this is what you should have learned from watching that video. So picture of model DNA from narration. So 1, the DNA unzips, the DNA polymerase, which is an enzyme, adds new DNA bases to each side, and step 3, when replication is complete, there are 2 new strands. So semi-conservative is when each new strand is made up of a new strand and one old. So when this DNA is split, you have half of the original and half new. So these would eventually separate completely, and this will be one strand, and this will be one semi-conservative, portion of it being new, portion of it being old. Key term, DNA replication. So DNA replication is the process of making an exact copy of a DNA molecule. So this process is DNA replication, semi-conserved DNA replication, which means half's old, half's new. Okay. Semi-conservative replication, the actual definition is each new molecule of DNA is made up of one old strand and one new strand. So remember, if you guys need more time to write some notes down, please press pause. As much as I wish we were in the classroom, we're not. And I know some of you write super fast and some of you write super slow and some of you use different fun colors. And if you need to press pause, press pause. Do what you need to do. Organize it however you want. Okay? DNA mistakes. So what happens when DNA is not copied correctly? Okay, what happens? Okay, so I was going to have you do activity 5, which is about mutations. However, it involves using technology that I have not taught you before and that I have not mastered myself. So if you are worried about activity 5, don't, because I have just decided don't worry about it. I don't want you guys learning new technology 2 weeks before school, just like I don't want to figure out new technology 2 weeks before school. Go ahead and skip that. But we're still going to learn about what mutations are. So mutations. So a mutation is an error in DNA due to a change in the nucleotide sequence. This can be beneficial, harmful, or neutral. Okay, so this means that there's something going on inside the DNA and it's different than the original strand. Okay, and believe it or not, it can be beneficial, though more likely it is harmful than neutral. Okay, so as you know, we've already done our study on evolution. So when species change, it's because of beneficial mutations. Okay. Okay. So we're looking at these dogs. So these dogs are called Whippets. As you can see, this Whippet looks very different than this one. So this Whippet right here is named Wendy. So Wendy has something called a double muscling, which means her DNA codes for her to have a much higher muscle mass than her lithe counterparts. You see how like skinny this dog is? So included are a few photos. So this is Wendy and another dog of the same species. And it just kind of shows how like your genetics can go. Now, just because she's like ripped, she looks like she goes to the gym 8 hours a day, that doesn't mean she's an aggressive dog. She's super sweet. She's just kind of the Hulk in dog version, you know. So frog mutations. So common frog mutations are not pretty, and they're usually the result of an area being polluted. So these frogs, as you can see, they all have extra limbs. It's kind of unsettling to look at. It's more disturbing when you know the source of their. So they are like this because of pollution in their home, whether it is, well, it's likely going to be human pollution, unfortunately. That's just what we do. So now we're going to get into the types of mutations. So a point mutation. So a point mutation is what happens when a single code changes. So for example, when this one changes, then it becomes a different letter. So now this is going to code for a different amino acid. That different amino acid can completely ruin a structure of a protein and can affect everything. So for example, if we change this sentence, the fat cat ate the wee rat, it would turn into the fat rat ate the wee rat. So it turns into a story of a cat eating something that it normally would eat to a not so great story about a fat rat eating one of its smaller relatives. So that does matter. So one thing about this is that this can be an instance where a point mutation can be neutral, because I don't know if any of you guys remember seeing that chart that went over the amino acids and the codes for each amino acid, but some amino acids actually have different sequences that code for the same thing. So I'm sure it's not as common as we would like, but what could happen is this can go to a different one. This 3 sequence can be a little different and it might code for the same amino acid. So sometimes there are point mutations that are harmless, as if it never happened. The other type is inversion. So what happens in inversion is a portion of the DNA sequence goes in backwards. So in this case, we have A, B, C, D, E, F, G, H, I, J. However, when it is inversed, A, B, C, D, E, F, I, H, G. So these 3 are reversed. They're inverted, okay, which is why it's called an inversion mutation. Another type of mutation is deletion. So deletion can be horrible because it can mess up everything behind it, like everything in the entire gene sequence. So if it deletes this, all of a sudden, the stop and start codons are no longer there. Okay, it can mess up so much, it's not even funny. So for example, if we were going to look at this sentence, so the fat cat ate the wee rat, and now the sentence is completely unreadable because one nucleotide was deleted. Okay, So box D, mutations. So mutations are most likely to occur during DNA replications. So the DNAs are moving into position and are not bonded together. The chances of a mutation occurring is one mutation out of 10,000 genes. So that is not per 10,000 nucleotides, that's per 100,000 genes. So 100,000 of these separate strands of DNA are made. There's going to be one error within that 100,000 genes. So that's a lot. So it's quite rare, but the process does happen so fast and mistakes do happen. So there are causes of mutations. So anything that can penetrate the nucleus of a cell and interfere with replication process greatly increases the probability of mutation occurring. Such mutation causes agents are called mutagens and or carcinogens. And some examples are radiant energy, stronger than visible light, such as UV radiation, X-ray, gamma radiation, such as radioactivity. So if you're getting an X-ray and you have cells multiplying within your body, it can cause severe damage. That's why the doctors always take precautions. Sometimes they give you a lead vest. The doctors go into a different room because it can possibly trigger some mutations. However, the risk is reduced for patients since they are only getting x-rays once or twice, maybe every few years, depending on how accidental prone you are. However, the doctors that are taking the x-rays are exposed to that hour after hour every day. That's why they have their own special building. They're usually behind a wall. Viruses. So viruses and chemicals. Chemicals can be environmental toxins, prescription drugs, or recreational drugs such as nicotine, vape, alcohol, cocaine. So environmental toxins. So we have the stuff that factories introduce. We have chemicals that we use to clean, makeup, stuff that we stick on our skin. That's why we have testing for all of these products to make sure they're safe and they don't cause any issues. We also have chemicals in overly processed food. This is just a funny. So I know there's a lot of discussion right now over organic produce versus other produce and fertilizers and all of the chemicals we spray on our crops. There are some that a lot of science has gone behind and proven to be safe, and there are other chemicals that aren't as safe. However, because they're older, they're more trusted, and they're encouraging for those to be used even if they're not as safe. Does that make sense? Kind of weird. I don't know. I'm not going to get too much into this because it's a whole world. But drugs. So drugs such as nicotine, alcohol, smoking, cocaine, etc. So these things can cause mutations. Specifically, if a pregnant mother is using these items, it can have catastrophic results for the baby. Effects of mutations. So this is going to depend on how does it change the protein, the type of cell mutation it occurs in, age and developmental stage. So when you are an infant and you are literally growing every single cell in your body, the risk of mutations when you're exposed to those things is going to be greater. I'm so sorry, I'm going to sneeze. Than when you're an adult who happens to smoke who isn't pregnant. Yes, you will still have mutations within your cells. However, they will likely be less catastrophic than if you're smoking with an infant. So this can cause birth defects or death. Children, adults, it can lead to cancer. I mean, cancer is not great, but it's less catastrophic than birth defects or death. Mutation, albinism. So we've already talked about albinism. Albinism is when your body does not produce melanin to pigment your skin and hair. Watch this video about it. It's very interesting. I can't play it because you will not be able to hear it, unfortunately. So beneficial mutations, so they happen, they are rare, and this leads to a physical change that is beneficial. So one of the main examples we've learned is Opposable thumbs. So opposable thumbs is the result of a genetic variation, which was positive in a species, and because it was positive, natural selection allowed that mutation to be continued to be bred into further generations, what led to it being common, and eventually led to us having that opposable thumb. And beneficial mutations is what drives natural selection. So another video on skid pigment. Please watch it. Appreciate it. So passing on mutation. So box H. So once a mutation occurs, it is passed on. So let's say there is a mother who has a faulty copy of a gene, and it happened when it was passed on to her. Well, when she has a baby, she has a chance of passing that on to her child, or the father has a chance of passing a gene that mutated in his sperm to a baby. Okay, so passing into cells. So you can only pass on a mutation to other cells if it occurs when the DNA is replicating to go through the process of dividing into new cells. And in offspring, this one's only going to happen if they occur in the DNA that's going to be in a gamete. So if you have a mutation in a cell in your arm, it's not automatically going to pass to your baby. Okay, it only happens if the mutations are in the DNA that are in the gametes. And that can occur because a mother is smoking or a father is drinking or they're exposed to certain chemicals. That can happen. Box I, universal code. So DNA is the genetic code. This is universal, and it's the instructions for making all proteins within a body. So the proteins are the building blocks that make an organism everything that it is. So if the DNA code has faults, then the proteins are going to have issues. So DNA and mutations are passed to offspring. So scientists can take genes from one organism and put them into another. So this is called genetic engineering. So genetic engineering is changing the genes of an organism to get a desired result. So possibly only because DNA is universal. So one of the most famous, probably one of the most dramatic displays of this, I don't know if any of you guys have watched the newest Jurassic World movie, where they were talking about creating the perfect dinosaur, and they spliced in DNA from frogs that went invisible and poisonous creatures and very aggressive creatures to create this mega dinosaur, which, as you know, went haywire, and he ended up hurting all the pretty dinosaurs. It made me sad, but that is one of the probably the most cinematic, dramatic example of genetic engineering. However, it does happen on a much smaller scale. So, for example, BT corn. So this is corn that has been modified to be resistant to the European corn borer. So that is a pest that can destroy entire crops. Next, this tomato. So this tomato was genetically modified to have it stay firm longer. So that way it doesn't turn to mush. Mushy tomatoes are the worst. Sun-up papaya is resistant to papaya ring spot virus. So that makes it it makes it healthy. It makes it more likely to grow healthy, to make it into the supermarket so we can eat it. Things such as plant viruses, they can wipe out entire crops. So the fact that this is resistant naturally instead of having to spray more pesticides, it makes it better. Innate potatoes, so potatoes have multiple enhancements. Arctic apples, non-browning. So these Arctic apples, they are not going to brown because their DNA makes it more difficult for that to happen. Golden rice is probably one of the more, it's kind of amazing, although it is very controversial in some circles. So golden rice is genetically enhanced rice. In some regions of the world, people only have rice as their main source of food. However, rice is not a well-rounded source of nutrition. So there are people going blind because their main source of food is rice. Well, rice lacks a nutrition called beta carotene. So that beta carotene, without it, they lose their eyesight. Some people even die from not having beta carotene in their diet over a long period of time. Well, scientists created this rice called beta carotene that costs just as much as normal rice. However, it contains that beta carotene and it stops children from losing their eyesight and dying from malnutrition, because even though they're eating the calories, they're not getting something that their body needs. However, people say because it's genetic engineered, it might be bad and it can't be trusted. So that's like a whole nother thing that I'm not going to get too far into. But kind of amazing. So fast-growing salmon, not a fan of fish, so this looks kind of gross to me. Moving on. Genetic modified ingredients. So the majority of genetic modified plants are used to make ingredients that are used in processed foods. So in the 1990s, food from GM plants were introduced to the United States food supply. 93% of all soybeans planted are genetically modified as of 2012, and as of 2012, 80 of all corn is genetically modified. So I know genetically modified sounds really scary. However, we have been genetically modifying plants for thousands of years. Only now we can do it more precisely with science. So box J, how is this done? So basically, you find a section of DNA that you want inside another organism, and then you take that, splice it out, and then you insert it into another organism. So it's often inserted using a virus to take that DNA and insert it into this, because that's what viruses do. Viruses take DNA, their own DNA, and insert it into a host. However, now we're inserting a beneficial DNA code instead of a harmful one. So box K. So, common example of genetic engineering for human insulin. So for example, we find the human insulin gene in a cell. We take that gene, cut it out. We cut open a plasmid, paste human DNA into that plasmid DNA. Then we insert the plasmid into a bacteria cell, and then that bacteria clones or copies DNA by letting the bacteria replicate. So now you have all of these bacteria that code for insulin, and they can now produce it. This leads to all bacteria that come from that cell will now be able to produce insulin. So what that means is that cell can replicate into 2 cells. Replicate into 4, 8, 16, 32, up until you have thousands and thousands of cells. Box L. So 3 other examples of how we use genetic engineering. So bioluminescence, so a glow from a jellyfish. So I actually did this one when I was in high school. We took DNA from a jellyfish. We spliced it into normal bacteria DNA, and then we grew this bacteria and it glowed, which was really cool. So BT corn, it produces its own pesticide. So it basically has fragments of this, it produces a little bit of this chemical that pests do not like, so they don't eat it, which is good because that means you can eat it. EnviroPig. So this pig normally eats soybeans, barley, other grains containing indigestible phosphorus, and this pig will excrete phosphorus that can run into fresh water and kill wildlife and contaminate the human wild supply. So, and it's expensive to monitor this. However, EnviroPigs have a combination of mouse and E. coli DNA in their genome, which prohibits, I can't even say that BTs produced in the salivary gland. So then the new DNA in viral pig pork is chemically the same as traditional pork. So the pig is fed the same grains. However, because it doesn't have this enzyme, it's not going to produce as much phosphorus. The phosphorus is capable of being broken down and it absorbs it, and then it's not going to be dropped into the water supply. Okay, just a funny. Now just relax your backbone and let yourself unwind. It's funny because this is the enzyme that makes it unwind. I'm sorry, G, but we're basically not a match, which is true. Those two nucleotides are not a match. So this is the last slide of the last doodle sheet you will do with me as freshmen. I'm so sad just thinking about it. I am currently working on the test and hope to have it up by tomorrow, Wednesday. So you guys can finish my class early if you want. Just know that I miss you and I'm so sad that the school year is ending the way it is. I would have loved to have been able to say goodbye to every single one of you. Sign your yearbooks if you want. But please, everyone, stop by and visit me at least once next year. I would love to see you. I know I'm still on campus, but it'll be different not seeing your faces every other day in class. But I hope you guys have a wonderful, wonderful summer. And just know that if you ever need anything, feel free to stop by my classroom or shoot me an email, and I'll do whatever I can to help you. Bye.